Hello everyone, and welcome to this video. My name is Wolf Woods. Well, not quite. That's close enough. Today, I'm going to talk to you all about a dog's world, the molecular evolutionary history of man's best friend. Speaking of dogs, here's a picture of mine. His name is Buddy, and besides being 9 years old and a rescue, and a really good boy, he's a Shorkie, which is a cross between a Shih Tzu and a Yorkshire Terrier. Both Shih Tzus and Yorkshire Terriers are dog breeds. But what exactly are dog breeds? A breed is a strain or type of dog that has been artificially selected for by humans. This selection has been for various traits including size, color, fur type, and even for personality traits like aggression or herding. There are over 450 recognized breeds of dog. To be considered officially a purebred animal, both the dame and sire, mother and father, of the dog must be from the same breed. Sorry, Buddy, you're a crossbreed, not a purebred like these fine dogs. Look at this wide variation in dog breeds. If you notice, all of these dog breeds pictured branch off from the same point, right here, which turns out to be only 200 years ago. This has led to domestic dogs being one of the most phenotypically diverse mammalian species. Unfortunately, this has also been linked to more heritable disorders than any other non-human animal. To understand why this is, and more about man's best friend, many scientists have performed detailed genetic analysis of domestic dogs to understand their molecular evolutionary history. Let's zoom out to the history of modern dogs. The closest ancestor to the modern domestic dog is the mighty wolf. No, not that one. That's the one. How did we get from here to good old Buddy doing whatever it is he's doing? At some point, a process called domestication occurred, where some wolves began to develop traits that made them less fearful and even cooperative towards humans. Over time, these bonds became stronger and reinforced by the emerging human population, with humans beginning to select the emerging dogs and breed them for certain specific traits. This event of domestication became an evolutionary bottleneck. But is a bottleneck? Well, it's this. Well, not quite. More seriously, a genetic bottleneck indicates a point at which a species with various traits and phenotypes, represented by the colored circles, is reduced down to only have a small amount of the population surviving to produce offspring, shown here. This causes the overall genetic variation to be drastically reduced in the future. In some cases, this bottleneck is caused by an environmental catastrophe, like a volcano. But in this case, it is simply that only a very small number of wolves actually made the transition into dogs, with those that did being interbred with each other quite a lot. All of this happened long ago before written language, so we do not have exact numbers on when this occurred, or how many times this occurred. The most recent occurrence is known to be at least 15,000 years ago by current estimates. This vast decrease in population size means that genetic drift can take hold. Drift is simply a measure of the sampling error when going from one generation to the next. Just by random chance, in a smaller population, an allele might be fixed, as compared to larger populations where this is less likely to happen. Thus, drift is majorly affected at population bottlenecks, and its effects can carry forward through several generations. This wasn't the only bottleneck that the dog population underwent. Remember those 450 dog breeds that we discussed? Well, some of these breeds are quite old. One of the older breeds in questions are Samoyed dogs from northern Siberia, which have shared a state from at least medieval time, though have been interbred with other populations because of their relative isolation from the rest of the world. However, the vast majority of dog breeds were developed during the Victorian period of England, from the 1830s to 1900s. This artificial selection of dogs was seemingly done for the reason that they could, out of curiosity about nature and how we shape it, as many of these dogs were paraded around in fancy shows, showing potentially the boredom that the era brought. Regardless, these two bottlenecks, from 1500 years ago, around to the time of domestication, and from 200 years ago, from the time of modern breeding, are defining evolutionary traits of dog breeds. These bottlenecks have led to a reduced effective population size, of which we can still see the effects today. But what is effective population size? Well, Effective population size is defined as the number of an otherwise ideal population which accumulates effects, such as those due to drift or inbreeding, at the same rate as the real, non-ideal population. If that didn't make sense to you, don't worry. It's a concept that, while fundamental, many people struggle with. Basically, it means that when modeling a population, you cannot simply just use the size at which the population is now, the census size. This is because it doesn't take into account the various life history characteristics of a population. For instance, say a bottleneck occurs. For a few years, there will be an extremely low amount of breeders in the population. Even when that population recovers, the offspring will simply not have the same diversity that the population had before the bottleneck, and will take a long time for the effective size to recover to somewhere even close to the census size. From these bottlenecks, a large amount of linkage disequilibrium is generated. Linkage disequilibrium is simply how often two genes or areas on a chromosome are associated with each other. If a population is in perfect equilibrium, every set of alleles would have an equal chance of being associated with a different allele based on their frequencies in the population. This is modeled on the left, where alleles A and B are found on entirely different chromosomes. When they are on disequilibrium, such as on the right, some pairs or regions will be further associated with each other than they should be. This can happen because the regions are close to each other, or because it is generated by having such a low effective population size for a long period of time. For instance, let's look at this bottleneck figure again. Initially, the pre-bred domestic dogs would have partially linked short-range regions on the DNA in their chromosome, represented by the colors in blocks. These regions were generated a long time ago at the initial domestication event, but have since been slightly broken up into the form that you see here. However, during the creation of breeds, only a few of these chromosomes would be able to be selected for, creating long-reaching linkage disequilibrium, as represented by the big colored boxes. In modern breeds, this leads to highly linked pairs of chromosomes, with some minor recombination able to break up the disequilibrium as seen in the bottom one, but not enough for anything substantial to be seen. As a matter of fact, this double bottleneck history for dogs has been verified by comparing the R-squared value, which represents how correlated any two given loci are, over various long-run distances between an observed linkage disequilibrium from within breeds of dogs and one generated by a model which had two bottleneck events, one from 10,000 generations ago and the other 50 generations ago. As you can see, the lines are heavily correlated, suggesting this theory has merit. You can compare the linkage disequilibrium data between within dog breeds and across dog breeds, with humans used as an outgroup. We can see that there's a significant increase in the linkage disequilibrium within dog breeds. The dashed line represents the threshold by which two loci would be considered unlinked. Both across different dog breeds and humans reach this threshold around 200,000 base pairs. To contrast, the within breed group only approaches this after 13 million base pairs. This suggests that recent domestication events have kept levels of linkage disequilibrium higher than they would be if domestication was the only bottleneck. Now, for these dog breeds to exist in the first place, intense artificial selection for specific traits occurred. This can unfortunately allow for selective sweeps or genetic hitchhiking to occur. This occurs when a beneficial mutation will arise on the genome, marked by the green square, and with low levels of recombination and high linkage disequilibrium can be selected for, while other mutations that are neutral or deleterious, marked by the red circle, are brought along for the ride. If selection, artificial or natural, is strong enough, this will spread through an entire population where all cases of the selected trait will be linked to those of the neutral or deleterious trait. Additionally, the inbreeding that is needed to maintain these purebred dogs greatly increases the chance that a dog may receive an allele which is identical by descent, meaning that it's from the same allele and the same ancestor which made its way back to the same dog. Buddy, what are you doing there? Sorry about that, I think he was trying to eat my homework. This again can have negative effects from the population, as it prevents linkage disequilibrium breaking down by preventing recombination, as if both alleles are the same, they would have no way to recombine to make a new allele, as seen in the bottom example, as contrasted to the top example, where the two alleles are able to be broken up by recombination. If this happens for a stretch of the genome, we see what's called a run of homozygosity, which is linked to the concept of identity by descent. An unfortunate example of this can be seen in poodles. Poodles have a link to a high copy number of KITL gene, a gene which is linked to increased squamous cell carcinoma of the nail bed. However, only black-coated poodles actually have an increased frequency of the cancer. It was found that there was a compensatory mutation on the coat color locus MC1R, which only the pale-colored poodles have. The light-colored poodles have a strong linkage to a compensatory mutation shown by the green square, which negates the effect of the KITLG high copy number. The dark-coated poodles, unfortunately, are linked to a mutation that does not compensate for the KITLG gene, meaning that the cancer is much more likely. By artificially selecting for dark-coated poodles, we have unintentionally allowed this negative allele to hitchhike, which unfortunately lead to increased rates of cancer in these poodles. Now, you may be wondering, how do inbreeding, artificial selection, and genetic bottlenecks interact with each other to create the dogs that we all know and love? Luckily, there's a few papers that deal with this exact issue. Here, we see a comparison of the 0 to 4-fold heterozygotic ratio, which is the ratio between amino acid changing heterozygotes to non-amino acid changing heterozygotes, as compared to just neutral heterozygosity. This showed that dog breeds have a lower amount of neutral heterozygosity, which makes sense for the smaller population size. However, they also contain a higher amount of amino acid changing proteins. This is hypothesized to be the result of slightly weakly deleterious mutations taking hold. We can interpret this under the context of nearly neutral theory. Neutral heterozygosity is proportional to the effective population size because selection is less effective at eliminating weakly deleterious variation in smaller populations relative to larger ones. Therefore, we see this negative correlation between neutral heterozygosity and the ratio of 0-fold to 4-fold heterozygosity. Provided there are enough amino acid changing mutations which are weakly to moderately deleterious, the population bottlenecks that domestic dogs have undergone have reduced the ability for negative selection to remove these deleterious variants. This suggests that most of the segregating deleterious mutations are more consistent with an additive model rather than a recessive. Despite what you might intuitively think, Marsden et al. In 2016 found that these effects were not causally linked to the inbreeding effects. This first diagram models what that previous graph would look like if there were high levels of inbreeding but no previous genetic bottlenecks. You can see that there's actually a slightly positive correlation slope as opposed to the negative slope that we saw previously. Next, we can see what happens if you remove identity by descent from your population by forcing a random association between chromosomes by sampling 2 chromosomes from different subjects. This would remove any effects of identity by descent and therefore remove the effects of inbreeding when modeling a population. As you can see, the correlation returns almost exactly as it did before. Additionally, if you remove the long runs of homozygosity, which are also correlated to inbreeding, you again see the return of this pattern. All this together shows that the recent increase in inbreeding effects don't majorly impact the non-synonymous variation within genomes, which lead to an overall decrease in fitness of dogs from the initial domestication event. However, the researchers still found that intense artificial selection for desirable traits lead to strong accumulation of deleterious variation of genes in genetic sweep regions due to hitchhiking. By selectively breeding a limited number of individuals during domestication or breed formation, you reduce the effective population size across the genome, which can have serious negative knock-on effects. The increase to genetic load due to weakly deleterious mutations is shown here to be primarily driven by ancient and recent bottleneck effects. Wow, that was a lot to cover. Let's recap. Looking through the evolutionary history of dogs shows 2 key bottleneck moments which highly impact the fitness of the species. Because the effective size is reduced so much early on, we still see some impacts on the weakly deleterious mutations on the fitness in our modern friends. From intense artificial selection to create dog breeds and the inbreeding required to maintain them, there are some unfortunate correlations between diseases and traits bred for that are intensified when the population is too heavily inbred, causing some alleles to be identical by descent and, when recessive, cause extra negative effects. This only occurs when the mutations are strongly deleterious. Weakly deleterious mutations, as shown by the nearly neutral theory of molecular evolution, actually care more about the effect of drift, has a larger impact on the two bottleneck moments that we talked about earlier. What's next for dogs? Well, besides avoiding heavy inbreeding, it appears that the best course of action is to make sure that there are large, effective population size to allow the population to recover from the effects of both the recent and ancient bottlenecks. Of course, further genome-wide studies were able to show specific polymorphisms which cause the diseases that plague them. Dogs have been extremely useful to study because of their wide array of phenotypes caused specifically through humans and artificial selection in the last few hundred years. This research might give us interesting insight into our furry friends and may pave the way into greater understanding of molecular evolution. Isn't that right, buddy? Good boy, buddy. Good boy. I hope you enjoyed the video. Stay tuned for part 2, where I just read off all of the 8 research papers that I did in a monotone voice directly staring at the camera. Till next time, I'm Wolf Woods.